talk a little bit about the preparation and properties of Esters lab. This lab, there's not a ton to it as far as the write-up that you actually have to do. I just want to, basically, I want to make sure that you understand what's going on here, what we're doing with these esters, and why we're doing it. And now, as far as giving you lab instructions here, this would have been useful if I had made it a little bit sooner as far as helping you out with the procedure. I'm just going to have to give you some detailed instructions when you come into class today as far as what you're doing. But essentially what you're doing here is you're going to take some alcohols and some acids, some organic acids, and you're going to mix those together. And then you're going to add a drop of sulfuric acid, and that's basically going to create this ester mixture. Okay, you put this in acidic solution, and these two things will combine. Sorry, excuse me, I'm fighting a cold right now. Anyway, so you're going to combine those two things, and you're going to make some of these esters that you see here. And you're going to get to choose which ones that you want to make. Some of these are made from things that do not smell very good. Which one is it? Butyric acid is just a horrible smelling compound, so we're going to keep that in the fume hood in the back, and hopefully that will keep the bad smell out. Most of the things that you're dealing with today should smell pretty good. But basically the general reaction here that they're showing you, the RCOOH, another way to write that that's going to look more like what you have on your sheet is R connected to C, double bonded to O, bonded to OH. Okay, and so you've got carboxylic acid there, and then you're combining that with an alcohol. Okay, and the alcohol is just the R group with the OH on it. Okay, and so what actually ends up happening there is you end up with a molecule that looks like this, and this is actually the way that an ester is written typically is going to look more like this. And then these R's just represent other carbons on the end. So you could have a you have a 3-carbon chain over here, 4-carbon chain over here. Doesn't really matter. And so they're labeling this with R prime to show that it could be different than the other R. But basically, you're just combining these two things here. Sorry. And then when you combine those two things, you end up with this. Okay. And then we're going to actually do the opposite in a because this works in an acidic solution. When we add sulfuric acid to this, it makes that. We can kind of go backwards with this as well, and we can do a what's called a destruction, or a what is it called here? Destruction of esters. I think it's also called saponification, but they're just saying that you're reversing the reaction here. So basically what you're doing on this one is you're putting an ester into sodium hydroxide, and then you're heating it up, and it actually breaks apart. And so when that happens, you're actually taking the reaction back this direction. So what they're showing here with this double arrow is that this is a reversible reaction. It can go either direction. And there are a few things on here as you read them. They're not going to make a lot of sense because we haven't really talked about reversible reactions or equilibrium or any of that yet. But this reaction happens both directions at the same time. And you can actually push it back so that it's going back to the left. And now you have your acid and your alcohol again. So you've broken it apart. And then it's not going to smell good. It's not going to smell like an ester anymore. So we're going to do that with methyl salicylate. Now, methyl salicylate is also known as oil of wintergreen, and you're going to recognize this scent because it's a scent that's in a lot of, any wintergreen gum or candy, any of that sort of thing, and it smells really good. So you'll be able to smell that at first, and then after you've taken it through the reaction, since you're destroying the ester, you shouldn't be able to smell it anymore after that. Okay, so that's the basic idea of the lab. It's a pretty easy lab, pretty fun, because you're just smelling things and seeing if you can make the smells that are on that chart. Down here, the post-lab questions, there really are no pre-lab questions, so all you really have to do is write a background section, record some data as far as what exactly you mixed and what scents you got out of those. But again, I mean, that's super easy. The only thing that's a little bit difficult on this lab are the post-lab questions. So number one, you do not have to worry about, okay? Because number one is asking about equilibrium and something called Le Chatelier's principle that we are not going to discuss until second semester. So you don't need to worry about that yet, okay? So just skip number one. The rest of these will give you plenty of trouble. It says using a chemical handbook, list 2 additional fragrant esters that were not discussed in this experiment. Discussed in this experiment. That's what that's supposed to say. 2 additional fragrant esters that were not discussed in the experiment. So you need to actually look these up. Find a couple that were not on the chart that we did in this lab, okay? Which shouldn't be that difficult. Number 3 is going to take you a little while. It says draw the structure of each of the esters listed in the table of common esters on the first page in the experiment. So remember, your esters are always going to have this general formula where you've got, some group, and then you've got the double bond, oxygen, and I'm sorry, not that. That's a carboxylic acid. It's going to look like that, okay? And so what you need to do basically is just, for each of these, draw in the R and the R prime, because those are the only parts that are really going to be different. This part in the middle is always going to be the same. So basically you have to do that for each of them, which is going to take up some space in your lab notebook because there's quite a few of those. I want you to also do that for the one that I drew in. Here, hang on just 1 second. Let me grab that real quick. Okay, so the one that I added here is the ethyl butyrate, which smells like strawberries, made out of ethyl alcohol and butyric acid. You should have that one on your lab. So you need to also do that one as well in your discussion here. Get rid of all this. Okay, so that's that one. Not hard, but it'll take you a little bit of time. Use another resource to write the definition for hydrolysis, so you just need to look up hydrolysis. Now, they do give you a definition in the lab. I think they just want a little more detailed definition. Let me see if I can find it here. I know it's in here. Here we go. So where it says destruction of esters here, part 2, I thought that's what it was. Yeah, here we go. Hydrolysis, okay? So the water and the ester reacting with one another to give an alcohol and an organic acid. You might remember me talking about this earlier in the semester, that hydrolysis is kind of the opposite of an acid-base reaction. So hydrolysis is a salt and water combining to make an acid and a base, okay? And so that's what's happening here with this hydrolysis. This alcohol is going to kind of act as the base in this particular instance, and then you have your organic acid, okay? Because you're starting with the water and the ester, okay? And the ester sort of works as the salt in this particular instance. So you just need to look up a good definition for hydrolysis, and then they want you to write the equation for the hydrolysis of methyl salicylate. So you're going to have to look up the structure again of methyl salicylate, or at least the chemical formula of methyl salicylate. And remember that the hydrolysis reaction is going to be the methyl salicylate plus water, and then you need to figure out what organic acid and what alcohol are going to be made when that breaks apart. Okay, so again, you're going to have to do a little bit of research on that one as well. And then number 5, again, going to have to do some research here. You may or may not have products in your home that have esters. Now, the way that you're going to know this is because they end in, if you look at the names of all of these here, Notice how they all end in ate, N-propyl acetate, methyl butyrate, isobutyl propionate, acetate. So you get the idea there. If you see that 8 on the end of it, then that is a hint to you that that might be an ester. Okay, and so what you're looking for is stuff in your house that that might be examples of esters. And where you'd want to look is in your shampoos, anything that has a fragrance to it. You know, soaps, those sorts of things. So see if you can find some things around your house that actually have esters in them. Now, if you're not able to find that, then you can maybe just look up some things. You know, what products contain esters, and then you could list those products. Let's see. It doesn't give you a specific. Oh yeah, it does. It says find the names of 2 esters among the ingredients. Okay, so you have to find at least 2, which is not that big a deal. You should be able to find those in your house. Okay, but it's just kind of an interesting activity to see. Oh yeah, so we actually use these esters to produce fragrances and products that we have in our homes. So anyway, fairly simple lab. The post lab is a little bit different in that it doesn't involve calculations. It just involves a little bit of research. But I think you guys are going to like this lab, and it's not gonna be too much work for you doing the write-up. So don't get used to that. We've got a couple coming up that are going to involve a lot more work. So all right. Anyway, that's that. Sorry if that discouraged you. I don't know why I needed to say that in this video. Anyway, we will see you in class.